Enzyme action, mechanism of action of enzymes, majorly two types. Enzyme action majorly two types. One is lock and key model of enzyme mechanism. Second one is induced fit model of mechanism. Induced fit model of mechanism. First we observed lock and key mechanism of enzyme action. See here it is in lock and key mechanism, enzyme containing their own specific sites. At the same time, substrate containing their own specific sites. The specific sites are called active sites. Enzyme containing active sites, substrate containing similar active sites. These similar active sites are involved in the formation of enzyme substrate complex. This type of enzyme action is called lock and key model of enzyme mechanism. Enzyme containing their own specific active sites, similar that active sites occurs on the substrate active sites, substrate structure. So these are similar substrate, the similar substrate structure of active sites present on the enzyme active sites. So they are cannot changed. The active site cannot changed and substrate structure also cannot change. This type of substrates involved with enzyme to form enzyme substrate complex. This type of action, enzyme action, is called lock and key model of enzyme mechanism. The next one is induced fit mechanism. Wantedly, the substrate induced to enzyme to change their active sites, to change the active sites of enzyme before the enzyme substrate complex, formation of enzyme substrate complex. Enzymes induced, substrate induced to enzymes to change the active sites, that active sites similar to the substrate structure by the induced mechanism. The substrate induced to enzyme by the induced method, that enzyme changed their active sites, that active sites are similar to the substrate structure. By this induced method, the substrate react with the substrate combines with enzyme substrate complex by the induced fit method. So these are the two mechanisms of enzyme actions. And mechanism of action is competitive inhibition. Competitive inhibition. That competitive inhibition, see here it is one enzyme containing their own active sites. That active sites cannot change it by enzyme. The same active site structure containing substrate. Before the substrate occupied the active sites of enzyme, inhibitors also similar to the substrate structure. Inhibitors also similar to the substrate structure before the substrate occupying of active sites of enzyme. The inhibitors are occupied active sites. So automatically what will happen? Inhibitors are occupied active sites. The enzyme cannot react with, or substrate cannot identify enzyme. Substrate cannot identify enzyme. They cannot form in the formation of ES complex. ES complex means enzyme substrate complex. So this type of action, enzyme action, is called competitive inhibition. Competitive inhibition means substrate and inhibitors are in same structure. If substrate occupied the active sites of enzyme, the ES complex is formed, then reaction will be started. If inhibitors are occupied the active sites of enzyme, then substrate cannot occupy the enzyme sites, so the reaction cannot will be start. This mechanism is called competitive inhibition mechanism. And next one is non-competitive inhibition. Non-competitive inhibition means substrate containing their own structure, inhibitors also containing their own structure. At the same time, enzyme also containing their own active sites. Before the formation of ES complex, ES means enzyme substrate complex, the inhibitor occupied other site of the enzyme, non-active sites of enzyme. Inhibitor occupies the non-active sites of the enzyme. So non-active sites are occupied by the inhibitor. By this inhibitor, the enzyme changed their own active sites. Inhibitor changed active sites. So active site changed means if substrate is cannot react with, cannot attach with the enzyme. So ES complex cannot formed in this non-competitive inhibition. The non-competitive inhibition sites are called allosteric sites. Allosteric sites. Allosteric sites means non-active sites. Non-active sites of enzymes occupied by the inhibitors. By these occupied inhibitors, the enzyme change their own active sites, but the substrate cannot identify the active sites of enzyme because the enzyme change their active sites. So they cannot form ES complex, enzyme substrate complex. And the next one is this allosteric effect. Similar, before class is completed, enzyme containing their own active sites. That active site changed by the inhibitors. The inhibitors are occupied non-active sites of enzyme. This type of effect is called allosteric effect. By this allosteric effect, the enzyme change their own active sites, so cannot involved in the formation of ES complex, enzyme substrate complex. These are the enzyme action mechanisms.